Good afternoon, friends. This is Dr. Mukesh Kumar Singh, and my presentation is plant tissue culture techniques. This picture shows complete programming of plant tissue culture. This is the content, plant tissue culture definition, and about the plant tissue culture. Second one is different techniques of plant tissue culture, importance of plant tissue culture, and name of books for reading about plant tissue culture. Now, introduction of plant tissue culture. First of all, I want to define the plant tissue culture. In plant tissue culture is the growth and development of plant seeds, organs, explants, nutrient media, trial or aseptic conditions. Now, what is the explant? Explant means the cut out a very small piece of leaf or stem tissue or even isolated individual cells and place them in a tissue culture container. It is known as explant. Or we can say explant means the any part of plant which is used to inoculate in artificial medium for regeneration of new plant. Now, first of all, we surface sterilization of explant, and after that it will be inoculated in artificial or nutrient media for regeneration. Now, different techniques of plant tissue culture. Plant tissue culture technique may be divided into two different groups. First, techniques for crop improvement. Second, techniques for micro propagation. In case of crop improvement, we can change the genetics of new plant, crop plant. These are some important techniques which are used to change genetics of crop plant. First one is development of double haploid. Second, somatic hybridization. Then embryo rescue technique, protoplast fusion. Double haploid can be generated with the help of anther culture, pollen culture, and ovule culture. In case of somatic hybridization, we can hybridize the somatic cells of different species or different genera. And now embryo rescue technique. This technique is more help for wide hybridization system. When we use two different species for hybridization or two different genera, it is known as wide hybridization. And in case of hybridization, there is no seed setting or mostly fertile. We can't get fertile progeny in wide hybridization, so we use embryo rescue to survive new progeny in case of hybridization with the help of embryo rescue technique. Now, protoplast fusion, fuse two different protoplast of different species or different cells, different organisms, and it will be regenerated in plant tissue culture. Now, micro propagation. Some techniques are used for production of clones with the help of plant tissue culture, just like apical bud culture, meristematic bud culture. Other plant parts like stem, root, leaf, and seed also used for propagation of clones. Now, importance of plant tissue culture. There are more there are several importance of plant tissue culture nowadays. It will be used in plant and animals category, and it is mostly used for genetic modification system. Just we know about genetic modified organism. It will be regenerated with the help of or propagated with the help of plant tissue culture. Plant tissue culture technique has the ability to rejuvenate the tissue rapidly. It produce exact copy of itself like as clone. It is a technique of quickly producing plants without any tubers, seeds, or bulbs. We don't need about any part of plant. We can use only single cell or tissue to regenerate a number of clones for a particular plant or animals. It also helps in conservation system of endangered species of plants and animals. It conserve biodiversity for long duration. Now, the summary. Now, I conclude the complete presentation of plant tissue culture. I have discussed about different techniques of plant tissue culture and system simplified of plant and plant tissue culture. It several use. How can we use plant tissue culture for agriculture and as a industrial level? Sure. Now, these are some important books for plant tissue culture. Introduction to Plant Tissue Culture, written by N.K. Rajan. It is very important book for make you understand about plant tissue culture. Plant tissue culture theory and practices written by S.S. Bhoswani. And these are the reference of my PPT presentation. Now, thank you very much.